lot faster, and maybe so the frequency of the genes for shorter legs might decrease. You know, if you had some gene that made your muscles be a little quicker, you know, so faster twitch muscles or something, so you run faster, those frequency of those genes might go up. Now again, we're talking about time. We're talking about time and a lot of interbreeding, of course. You know. That just happens in one individual, but it's not affecting the entire population. So we're talking about having those genes affect the entire population. Am I making any sense here? You guys just sort of seem to sort of. I wonder.
It wasn't that. I panicked because I accidentally hit the button and stopped the video.
Oh.
I just screwed us all.
And you fell asleep
No. Okay. That happens just about once a class, but it hasn't happened yet.
All right. Okay. So anyway, so let's talk about the certain kinds of things. We distinguish between, I say we, you know, the people who deal in this. It's not my field, but that's okay. People who deal in evolution distinguish between what we call micro evolution and macro. Micro meaning small, macro being big. Okay. So in microevolution, we're talking about, let's actually write this up here. So microevolution, we're talking about the change in gene frequency over a relatively short period of time. You've got to be careful about this, you know, using that word time. When you're talking about evolution, and you're talking short time versus long time, a short time still could be 100,000 years.
That's what I thought, yeah.
You know, you've got to be really careful. Long time might be, you know, 2 or 3 million years. So you've got to be careful. I mean, we're not putting time things up here when we're saying short time, long time. There's no official definition of the amount of time involved. It's you know, a short time is relatively short time
compared to 4 billion
years, maybe
4 billion
thousands of years or something, as opposed to several hundreds of thousands or more likely millions of years for macroevolution. Yeah.
So then the changes in the pigeon population is a low scale adaptation.
Yeah.
Not an evolutionary trait.
So what is the change?
They're flying higher than they should be able to, and they're nesting on buildings that they have no way to be able to get to.
This is an unusual. This is
an unusual thing that's happening in the past 20 years.
So some change.
So they're changing and affects us.
Right now, but some change. So are they saying some change in their wing structure that allows them to fly higher?
They're saying that their wings are bigger, they're larger, longer.
So that would be an example of microevolution.
They said in the past 20 years.
Oh wow! That's a good short amount of time.
They're finding pigeons up on like the top of I forget the name of the tower.
Yeah, but some
pylons coming out the top. Wow, they're finding pigeon nests.
They never up that high before. Huh, interesting. Yeah, and now that's a really short time, and that still fits in this. That's why I say a short time. You know, we could be talking for some things like this, a short time. Well, the way some of the bacteria change. Yeah,
that's a
it makes a difference.
That's environmental effects. That's different.
For a long time.
Oh, and not being seen. Yeah, maybe it's just been noticed. That's possible. Or for whatever reason, I mean, you had those guys may have been around already, but it wasn't until recently that there was enough competition for the lower roosting sites that they decided to go higher
to another niche. Yeah.
So yes, but it's interesting if they actually got some pigeons from before that and checked out some of their genes and checked it out.
These could have just been things that they were expressing earlier until they had
or not noticed or something. And maybe just a behavioral adaptation. Who knows? I don't know. It's a good question. I don't know anything about these guys, so I couldn't say. But there's several possibilities, and one could be just a change in some specific genes and changing the frequency so more of them have the genes that allow them to get up there. Yeah, could be. Yeah, well, megalosaurus.
What about the ability of humans to eat lactose after the age of 2? They're saying that we only have lactase up to age 2. And then
here's the deal. I will tell you quickly about this. The lactose intolerance. Mammals. Mammals. What distinguishes mammals from other vertebrates is that they make milk, have mammary glands, and that's why they're named mammals. They make mammary glands, and so the young are fed on mother's milk. Mother's milk, we're talking mammalian milk, has in it lactose as the sugar, the main sugar. And lactose is a disaccharide. It's a combination of glucose and galactose, and they're hooked together. And in order to use it for, as a sugar source, excellent sugar source, you've got to break the bond between the glucose and the galactose. And we have the enzyme lactose. All mammals make this enzyme called lactase, that breaks that. Then you use those individual sugars for energy. It's great. But essentially all mammals, I said essentially, so I'll come back to that word, but essentially all mammals, after they've been weaned, they stop making the enzyme lactase. They still have the genes for it. You still have the genes. You just stop making lactase, and from a practical point of view, why waste your energy making an enzyme you're not going to be using? Because if you've been weaned, you're never going to see milk again. If you're a horse, you're weaned, you're eating grass and hay and stuff like that, So you're not ever going to drink milk. Some human populations around the world kept making that, presumably they kept making the enzyme even after they were weaned, and those are the populations that eat milk products, throughout their life. The other populations that don't make it, they eat milk products. They get all this terrible digestive kind of upset. They don't like it, so they stop eating it. It's a fractured kind of thing. Why some populations keep making the enzyme and others don't, no one really knows. But it's some kind of a control that, and maybe it's a control gene that got changed. Not the lactase gene, but the control gene that says turn it on or turn it off. Maybe something like that. By the way, lactose intolerance, what happens when you eat lactose, milk products, if you have so-called lactose intolerance, is it gets through your stomach, gets into your intestines, and suddenly in your small intestines, or actually your large intestine, I should say, because normally milk gets digested in your intestines, your stomach, but mainly your small intestine. But you absorb all the sugar. So rarely does sugar get into your large intestine. It suddenly gets into your large intestines, sugar, because you weren't able to break it down if you have lactose intolerance. And you have all kinds of bacteria in there and go, yes, lactose, we haven't seen that for a long time. And they start eating that lactose as fast as they can. And as they eat it, of course, what do? They give off acids and gases. The acids make it irritating to your large intestine, and causes things like diarrhea. And the gases, well, you know what gases cause.
Methane.
We won't discuss this in polite company. Oh, well, this is biology one, I forgot. It's not polite. But anyway, but and it's the same thing in beans, by the way. Why beans cause so many great problems? And it's because there's a triple sugar in there. There's 2 triple sugars in the beans. One's called raffinose and the other's called stachyose, whatever. But they're triple sugars, and and we don't have the enzymes to break apart those sugars so we can use it as energy. But some of the bacteria in your gut do, and when they get that, they go, yes, stachyose, raffinose, good stuff, and then they break it down. They make acids and gases, and we get all those good effects from it. But anyway, but back to your point. I think what was your question? Why do some people do it?
Certain populations of humans have micro-evolved.
And maybe that's it. I mean, maybe they micro-evolved with, and I think it's probably a control gene.
Because they're around large animals and
Yeah.
they have access to milk.
That's what it would imply.
They
Yeah.
lose the ability to have lactase and then re-evolve this, or evolve this lactase through adults.
Yeah, and it's a good question. And as a hunter-gatherer, you wouldn't usually be exposed to milk products. It's only, as you said, if you stop and you are raising, sheep and beef and stuff.
That are lactose intolerant, if they were exposed to milk long enough.
That's an interesting question, but it's hard to do that because it might take 1,000 years, so it's sort of hard to run the experiment and find out, But that's a good question, and it could be that those populations that naturally kept making lactose were the ones that went ahead and they tried it and then they kept eating it, or it could be that they micro-evolved, such that there was maybe a mutation in the control gene, so they kept making the lactose. That's got lactase in it. That's kind of what I think, but I'm not sure. I don't know.
I read that it actually is a mutated gene that keeps us to make us be able to digest milk, and people who are lactose intolerant don't have that.
Do they mention if it happens to be like a control gene to turn it on or off? Turn off the.
All I know is that.
See, my guess is it's a control gene that and if you in one form that says
make
go
it.
ahead and transcribe the messenger RNA off of that lactase gene, and if you don't have that, it says don't transcribe.
Yeah.
That's what I think, but I'm not positive. But that's what makes sense to me, but I don't really know for sure. Interesting stuff. Oh wait, Anand, you had a question.
A lot of modern day lactose intolerance isn't genuine lactose intolerance. It's an intolerance to the homogenization of fat in milk.
That's possible.
And a lot of it's based on blood type too, as your tolerance to just meat or fat.
It is more complicated. What you're saying is.
It's all a huge thing. People say you should eat meat or you should be a vegetarian. I'll die if I'm a vegetarian.
Supposedly had.
I have hyperthyroidism. I just simply can't do it.
But he said he had an allergy to cow's milk, but he could drink sheep's milk or goat's milk, I guess it was.
Because it's naturally homogenized.
And that may be the thing. So anyway, so some of but and I think you're absolutely right that not all of what people think is lactose intolerance is actually could be a problem of something else. But anyway, whatever. So but these things are complicated. Was there another question? No? Okay. And depending upon how much it's you had a question. Did you want to answer that? Oh, okay. At any rate, so when they talk about microevolution, we're really just talking about change in gene frequency over a short time, and usually we're not talking a huge change in gene frequencies, just a few genes. When you're talking macroevolution, that's a different story. Macroevolution is a long period of time. We're talking about sufficient change in genes or gene frequency that you actually develop new species. And that we'll talk a little bit about that. So I'll say enough change in gene frequencies.
Question.
To make a new species. And generally speaking, we're talking a long period of time. Now, again, long period of time. We're talking maybe 2 million years or something like that. Maybe who knows what.
I mean, how old are multiple celled organisms? Are only like 700,000.
Wait,
multiple celled.
Yeah, about 600 to 700 million.
Is that what it is? Million.
Million.
Okay. Did I say 1,000? Yeah.
So
million. Yeah.
Somewhat less than a billion years that we've had multicellular. They figure, but and so, but macroevolution, you can figure just the most recent ancestor between humans and chimps was about 7 million years ago. So somewhere around 7 million years ago, these two groups split off from the ancestor, and one developed into the modern chimp, one developed into the modern human. It wasn't a quick development into modern human because modern humans, we've only been here. I haven't been here quite this long, but modern humans have only been here about roughly 250,000 years.
15 seconds.
This development of all these, what you want to call it, proto-humans or whatever term you want to use for them. So I'm not saying it's an immediate thing to modern humans. I'm saying the branch. And that's another thing, by the way. You've got to be really careful when you're talking about evolution because we often like to look at it as just this organism to this one to this one to this one. Oh, and here I am, right here. This is us. But evolution is a branching thing. It's more like a bush. If you go out and look at a bush and see all these branches coming out, that's really what has happened over the millions and millions and millions of years. Is all this branching and
recombination too,
dying
right?
off, and then but here this little twig right here and this little leaf right here. Hey, that one's okay. That little branch right there is still around, and this is some particular organism. So keep in mind, it's a complicated thing. It's not just a simple pathway directly from A to B to C and all that. There's all this branching. Some of them survive, some of them don't. And obviously the vast majority don't survive. Some of us are here. I don't mean us, but I mean organisms on Earth are here. But far more have probably died off. Far more have died off than are still alive today. But anyway, so in terms of just those words, macro and micro, just keep in mind the macro is what's referring to evolution of species, and that's a long involved process. Let's talk about microevolution, just the change in gene frequency. Let's talk about this, and talk about what I have up here: 5 ages of microevolution. And we've talked about these, some of these already, so it's not like this is totally new, but some of them we haven't spoken of specifically. I'll talk about that. So we'll say, well, I'll just leave that term to this time. So we'll say 5 agents of microevolution. And one, we've talked about already is mutations. And what I've said before, but I want to kind of stress this, because we've talked about mutations, and mutations will cause changes. But when you're talking about organisms that are evolved at this particular point, we could be talking about fishes or humans or plants or whatever, bacteria, whatever it might be, they're already fit for their environment. Okay, they're doing quite well, presumably. As long as the environment doesn't change radically, they're going to be just fine. So in that case, on the average, if a mutation came about, it's more likely to be bad than it is to be good. You know what I'm saying? It's more likely to have a harmful effect. Or, I mean, remember that a lot of mutations are neutral. They don't have any particular effect at all. But on the average, you expect a mutation to have, if anything, a slightly bad effect rather than a good effect, because you're already fit. Make a change in how fit you are, and it's probably not going to be good. But some are good. Some might have some beneficial aspect to it. Or some might just be a variety of variation that in a different environment might be useful. I want to talk about that idea just in a second. I want to answer a question, but talk about that in the sense of things like the sickle cell anemia. Question, you have questions?
You just explained what you
Oh, say it, I'm sorry.
You answered my question.
Oh, okay. Okay. So here's the thing. As an example, there have been many mutations in hemoglobin genes over the years. Most of those mutations are bad because hemoglobin is the carrier of oxygen, and you need this hemoglobin to carry your oxygen throughout your body. Okay. And so, but they found people with various kinds of problems, and they've looked at things. They've looked at their hemoglobin, and they said, oh, this is. They call it a hemoglobinopathy. It's some problem in your hemoglobin. And most of them, as I said, aren't good. But most of them, they may not be good, but they probably. I mean, a severe change in your hemoglobin where you're not carrying oxygen at all, well, you probably aren't going to survive. Okay, so that's bad. But you can have some little small change, like a point mutation. Remember we used the word point mutation, like 1 amino acid difference from what the majority of people's hemoglobin is like. That usually won't have too much of an effect. It could have a tiny effect, or in some cases it could have a big effect. And the example of the big effect was sickle cell anemia or thalassemia. Those are two slightly different point mutations, but they both cause a slight decrease in the ability to carry hemoglobin, but not deadly usually, except in double recessive homozygous form. But it turned out, of course, that, as you know, we've talked about it before, that those mutations give you a slightly beneficial effect for survival if you're around malaria, because malaria is a nasty disease, and it kills from 10 million to 100 million people a year. I mean, a lot of people. Over 10 million people a year die of malaria. And if you have a mutation in your hemoglobin, it gives you a better chance of surviving malaria than your neighbor over here who doesn't have it, then you're probably going to survive and you're going to pass on that particular gene. In the double, in the homozygous recessive state, it's deadly, but in the heterozygous state, it gives you survival advantage against malaria. So it's not a good gene to have in an area without malaria, but in an area with malaria, it's useful. And that turns out just to be both of those, thalassemia and sickle cell. Both are just one single mutation, they're different mutations in the two diseases, but they're just one mutation of one nucleotide causing one difference in an amino acid that's put into that hemoglobin, which causes a little bit difference in the structure. Anyway, so it's weird. So having that gene for the sickle cell, in the population, in this case, was useful in certain areas of the earth. But anyway, so mutations are sometimes said to be the raw material, or often referred to as the raw material of evolution. Because they provide variation. And again, I just want to say it one more time. Don't think a mutation is automatically bad. Don't think it's automatically good. You don't know. If someone says, "There's a mutation of such," you don't know how it's going to be until you try it. In other words, see what the phenotype is and see what happens to the phenotype. But if you just suddenly made a mutation in an organism, who knows what's going to happen? To be honest, not honest, but to tell you a little history, a little molecular biology history. Remember I said that the easy definition of a gene is a gene makes a protein, one gene determines how to make one protein. Well, that idea originally came from the 1930s. A couple of people did some experiments with yeast, molds actually, fungi, and what they did is they took a bunch of bacteria and they would zap them with a bunch of X-rays, and then they'd say, "What are there any changes in what these?" I said bacteria, yeast. These are not yeast, molds. Start over again. Fungi, molds. Okay, so they took a certain kind of mold, Neurospora crassus, and it was a black mold, the kind of mold you see on bread if you leave it out. And anyway, they irradiated these. They'd have a whole bunch of things they irradiated. And then they say, I wonder by hitting it with, radiation causing mutations, are we causing any of these colonies to lose some ability? For example, maybe they're losing the ability to use some particular food source that they could use before. And they would select for that. They check and see. Sure enough, they could find various groups of these molds that couldn't do certain things. And then they did a whole bunch of studies, brilliant set of studies, by the way. And that's where they came up with the idea. What these molds were losing were certain enzymes, the ability to make certain enzymes. And these were enzymes that were needed to break down certain food sources or whatever they were looking at. And so they're the ones that came up with the idea of what is a gene? A gene, one enzyme is the way they describe it. What is a gene? A gene is the information to make one enzyme. Well, an enzyme is a protein, and there are a lot more proteins than just enzymes, a lot more functions of proteins. So now that's why we make it really general. One gene, one protein. It's the information to make a protein. But anyway, the point is, by just zapping it, causing mutations, you can say it doesn't cause some change in the phenotype of these fungi. And the answer was yes, and they could actually select for it. Started the same. Yeah.
This probably doesn't. I mean, it does. Since we know that enzymes can bond molecules, right?
Yeah, enzymes that will put molecules together.
Do we use those to try to bond molecules together?
Oh, you mean do we use them commercially or something like that? In some cases, yeah. They actually will. God, when I was 30 years ago, they were actually putting enzymes in detergents. You could buy detergents that with enzyme action, and they put enzymes that would break up some of the molecules that stain and
The blood and the
stains
protein and
stuff
all
like
that stuff.
And they put these in. The problem was that there were a lot of people that were getting allergic reactions, just skin type reactions to all these enzymes on their skin, so they stopped putting them in. But for a number of years, they had these big advertisements, all these enzymes, and they put a variety of enzymes in that would break up things like blood proteins.
Yeah.
Help clean your clothes in case you had all these various stains.
Okay.
Yeah, but I don't, and commercially, you can buy, like, especially if you're doing research, but I'm sure all kinds of other things. There are companies that will purify certain enzymes to be used for certain purposes. And I think the enzymes that they got that they put in the detergents were probably bacterial enzymes.
Yeah.
And the people were probably reacting to some of the bacterial products that were still in them. Soaps with the detergents. But anyway, whatever. Okay, so let's talk about another thing, what they call gene flow. Remember, these are agents of microevolution. And microevolution is change in gene frequencies, over some, relatively short period of time. So gene flow. And this is just referring to how genes can move from one population to another. Now, I say genes, when I say a gene is moving, obviously it's the organism that is moving, and the organism has the genes. But you're thinking about, you have two organisms, the same species, but they've been kind of living apart, and maybe we have certain genes in this one group that's a little different from this other group, and then suddenly those groups come together. Then we can suddenly have genes from the two groups intermixing. And so some of the genes that this group has that this didn't can suddenly appear in this other population. That's what we're talking about, gene flow. So it's a movement of genes. Movement of genes from one population to another. We'll say, for example, by migration. I'm thinking of animals or plants, where they're suddenly, they may have been separated, but suddenly they're getting together for whatever reason. Okay, so migration. And then these two populations are intermixing, and so you're getting an intermixing of the genes that they have. Okay, here's an interesting one. Let me see if I have that book. I'll do it over here. Number 3, genetic drift. Genetic drift. The idea of genetic drift is how you can have some kind of a chance effect causing the gene frequency of a population to change. I'll give a couple of examples. Say a chance change
like
gene frequency in the population.
Climate change, like sudden or something like that.
We'll do this instead. A chance change. That's what they say. A chance change in gene frequency in a population. In other words, you have a population that has this, kind of a random distribution of the genes, and then something happens to change that gene frequency. So I'll give you a couple of examples. One's called a bottleneck effect. Now, you think of a bottle with a narrow neck. If you're trying to pour something out, it has to go. All that water or whatever can't get out of that bottleneck as fast as it could. You have it more open kind of thing. That's where they're using the word. But what we're talking about here is, some change. But this is chance change. Let me give you an example of the bottleneck effect. The elephant seals, those elephant seals have this funny big nose and they make that huge amount of noise and stuff like that. They were hunted almost to extinction about, 100 years ago or something like that. Almost to extinction. I think it ended up, and this is the northern, I think it's the northern elephant seal, if I remember correctly. Anyway, but anyway, they're hunted almost to extinction, and it ended up before they were protected that there were like 20 individuals. That's all, 20. Okay, and so, but then those 20 have been protected, and they've been breeding, and so the populations come back up, and I don't know how many there are now. Thousands of them.
Is there enough genetic biodiversity?
That's the problem. And that's what this bottleneck effect is saying. You presumably those 20. Now, I mean, there had been probably hundreds of thousands of these elephant seals. So there's a large gene pool, and then suddenly you're down to only 20. Are those 20 representative of all the genes that were available in the gene pool?
No,
Probably not.
no,
Probably they have some select number. Not that it's any special number. There was a chance which individuals were still alive. But the point is that you only have 20. There's not very much variation, and so you have what is they call, I mean, this is the term, bottleneck effect, because this is definitely a chance change in gene frequency in the population. So let's say if a population dwindles, and this example that I'm giving is that elephant seal dwindles to a few individuals.
Then sharks will die.
And then rebounds. It's probably not a random distribution of genes in that population now. It's a much smaller range of what genes are available. So the gene frequencies have definitely changed. And what are we talking about in microevolution? Change in gene frequency. So probably there are various genes that aren't even represented anymore because by chance they weren't in this little tiny population before they rebound. Let me just write this down. So if a population dwindles to a few individuals and then rebounds, the remaining, let's say the genes in the population now are probably a lot fewer than what was available. The genes in the population now, or I can say, are probably not representative of the original population. So this is called the bottleneck effect. And I think this is probably what did happen to cheetah. I mentioned the cheetah to you once before, I think, of having, I mean, every cheetah in the world is in a captivity of some kind, and there's a fairly small gene pool. There's not a lot of variety in the gene pool of cheetahs. And I think it happened, I don't know exactly how it happened, whether they just killed off a lot and they have, but I think, again, it's the same problem with bottleneck. Yeah.
Humans are bottleneck sometimes too.
Yes.
Those of us of European descent, we haven't survived the plagues, which wiped out whole
yeah, no, you're absolutely right. That is a good point, that there have been several events that have wiped out huge numbers.
Those of us who have Native American blood, been bottlenecked.
Absolutely. Been so that's absolutely true. There's been a lot of events that have caused bottleneck effects in humans. A lot of it, some of it was just diseases, some of it was genocide. Obviously, yeah. No, absolutely, that's very true.
There's one that they say that they could find in our genetics back like 12,000 years ago or something, when a lot of the mammals on the planet disappeared.
Oh. But that was from. But they think that, what do they call it? Oh, I'm not sure. I was thinking that your thing was just the fact that you're saying a lot of mammals disappeared, but that was, I thought they were thinking that humans were getting to be too good at hunting, but maybe that you're thinking of a different thing. I think. But yeah. But you're right, absolutely right. There have been various bottleneck effects in humans. Did that knock out a lot of our variations that may make it that we're less able to survive some insult in the future. Who knows? I mean, we don't know. As I say, a lot of these things we're only talking about. We don't only give ideas. We don't know unless something happens.
But I was just wondering, like, okay, so now they're down on one. You're serious? All the cheetahs left are in captivity?
Yeah.
All of them?
Yeah, I think that's what they say. And some of them are like wild animal parks, so they're not in a cage.
Well, I know.
But
I get
but
San
they're
Diego,
all-
but
there aren't any running around wild in the.
my follow-up question to that is, if their say is only 20 or whatever, over time, even if it causes problems, is that enough for crossovers to occur over, say, what we would call short periods of time or thousands of years to re-establish them as a species?
Well, no, you mean if they didn't die out? I mean, are you saying
If they're bottlenecked to a really small number,
of gene? I mean,
what,
very low gene.
can that low gene pool eventually stomp out the species?
Oh, that's always the worry. That's always the worry when you have very little variation. Something could come along like a disease, and they'd all die off. I mean,
Because they don't have
and
any.
if you're down to a really small number, it may never rebound.
Yeah.
But again, if you have things like maybe over a period of 1,000 years,
Yeah,
or 10,000
mutations
years,
could
enough
make it
mutation,
happen.
it'll get
Okay,
more variation.
Okay.
It might come back. I mean, this was the worry. I mean, you know, the American or the California condor, I think it's a magnificent bird, and it almost died out. And the other one that almost died out was the whooping crane. And they've been working to reestablish them, and I hope they do. I really hope they do, because you know, magnificent animals. But the worry, of course, there's various worries. And one is there's an awful lot of money put into just surviving, you know, allowing a few animals to survive. I'm glad they're doing it myself, but there are a lot of biologists say no, we should be putting that money into helping, you know, habitat, blah blah blah. Well, and that's fine. I understand that argument. But what my point is that by having so few, and yes, you're trying to interbreed, but still, it may be because of the bottleneck effect, it may be difficult for them to survive in the future. Who knows? We don't know. I mean, we hope they can make it. We hope there's enough variation so that they can make it.
Trying.
No matter what insult comes along. Yeah, go ahead.
We're actually watching videos of what happened in Polar Bear right now.
Oh, dude, yeah.
That's right. They're dying off.
They're drowning.
South, interbreeding grizzly bears.
Yeah. We'll have some pretty wild animals coming along.
Rather large and aggressive ones, yes.
Yeah.
I think another argument that people like make against like trying to help species that are dying off along, but you're kind of messing with evolution.
Yeah. But a lot of the reason for them dying off is because of us in the first place, you know. I mean, we have just messed up the habitat so badly. So you know, some people say, let's see if we can reverse some of that. No, I understand a lot of these counter arguments, and
The determinist argument would say,
and-
well, yeah,
and on the one hand, another argument is you say, look,
stopping it is part of evolution.
extinction is a natural fact. I mean, far more species have died off over the millennia than have, or the eons, I guess we should really say. Far more species have died off than are present today. So it's actually a normal fact of existence on this planet. But from a practical viewpoint, living in the here and now, you say, gosh, we're about to destroy all the condors. I myself think, God, that's a shame. It's a magnificent bird. I would hate to see it happen, But anyway, so I can't tell you what to think. I mean, I want you to think about these on your own terms and however you look at it. The financial aspects are one aspect. What you're saying about messing with playing God is what some people say. But we've been doing that anyway. I mean, the problem, of course, I mean, I said this so many times, I'm glad I'm a human, but my God, we're destructive. We're tough on this planet and on other species and on each other and whatever else you want to say, I think it's just chance that we're so effective at these things, and we'll talk a little bit about that. I think it's next lecture we're going to talk about the evolution of humans, and I'll talk about the Great Leap Forward and all that. So who knows? But anyway, let me tell you one other example of genetic drift. I mean, there are a lot. These are just some examples to think about. There's one called the founder effect. Founder effect. And here we're talking about the founder of a population. You might have a group of organisms that break off from the main population. Here we were, I guess I erased it out, but we were talking about the population. You have this big population of interbreeding organisms and all that. But it could be that you could get a small group of these individuals leaving that population for whatever reason and going to a new area. Okay. So now, and they're not coming back, they're not interbreeding with the original population. They're on their own now. And it could happen by chance. It could happen that a few lizards fell onto a log on the coast of Peru, and that log floated out of the Galapagos Islands. I mean, so it could be a chance kind of thing. It could be a deliberate kind of thing where some. And I remember seeing this movie about the wild dogs in Africa, and God, they can be some rough things to watch sometimes. But at one point, they showed this one female who was subservient to the main female of the herd, and I think, I can't remember if she finally did it, but she was trying to break off with some of her babies and another male and leave that herd of wild dogs and start her own. And presumably they weren't going to come back and meet. Well, anyway, but the point about this in terms of the founder effect is that the group that starts this new population is not necessarily representative of the entire population in terms of its gene frequency. So again, you're starting with a group. What are we talking about? Change in gene frequency. We're starting with a group, but it's a certain representative sample of the genes in the population, but certainly not a random sample of all these things. So it's just a way that you can get a group of organisms over time, short period of time, with a change in gene frequency. So all the relatives of this founder, these few founders, will have just a limited number of genes with respect to what the original population had. Let me write this down, and then I'll get your questions. Hang on just one second. So the founder effect, say if a few organisms, or individuals, I don't want to say organisms, individuals in some population. Actually, I'll give you a human example in a minute. Individuals in a population start a new colony, I'll say colony. Their gene frequency is probably more limited than the gene frequencies in the population. They probably, again, all this is just hypothetical. A different gene frequency than the main population. We have a different, I'm not spelling all these words out, different gene frequency.
That's okay, I'm recording what you're saying.
Okay, compared to the main population. I can't write that down. So these are just examples of how microevolution can occur. Just a few kinds of things. And there are other, geneticists will give you, or evolutionists will give you all kinds of other ideas too. Change in gene frequency. Yeah.
Like the difference between like the animal tiger versus Siberian tiger versus.
And yeah, but I don't know what the difference. I can't remember the differences about them. But yes.
Some of them are smaller.
Yeah.
Or just like Asian and African
some
elephants.
But I was going to give you a good example of founder effect that is present today in the world. The Afrikaners in South Africa. They all descended from a relatively small group of Dutch people who immigrated to South Africa. Mm-hmm. But in that group of people who migrated, there happened to be quite a few of them who had Huntington's disease. Remember we talked about Huntington's disease, Huntington's chorea, and we talked about Woody Guthrie and Arlo Guthrie. Mm-hmm. And that happens to be a dominant gene, by the way, but the effects don't show up until you're like 45 or 50. So, by the time you've had kids, you don't even know that you have it. It's not until later. But anyway, so it turned out that so in the founding group who went to South Africa had, several of them, I don't know how many, had the gene for Huntington's. They didn't know it until later when they came down with it, but then they passed it on to their kids. But then, of course, it's a small group, and they intermarried. And so the incidence of Huntington's disease among the Afrikaners is far higher than it is in the general population of everyone else. Does that make sense? Again, founder effect starting with a limited number of genes. So the more, those few genes are intermixed, but there's not really much variety. So far, okay? You guys with me? Yeah? I'm gonna open the door.
I say I haven't fallen asleep yet. You're good.
Hang on just 1 second. Thanks, Mark. Thank you, giving me a nice day.
No, it just looks that way, and then it gets windy and cold.
Well, it's a cool breeze. Okay, another aspect of we're on the we're got to number 3. Let's go to number 4 then. Number 4 is sexual selection. So what are we talking about? We're talking about agents of microevolution. Agents of microevolution. What are some things that will cause changes in gene frequency? Okay, sexual selection. Now, you've all seen those pictures of the birds. The male, usually the male bird is, really decked out. The female bird is a lot less flamboyant. And something about the flamboyance of that male, like the peacock. The peacock can, have these fancy feathers and stuff like that. And presumably these fancy feathers are what attract the female. So, because of sexual selection, there can be, a change in gene frequency over time, because the female in this case can just choose those males that have whatever, in the case of fancy feathers or whatever it might be. The bowerbird, some of you have seen the bowerbird, they make this fancy kind of a nest thing. But so again, just a few of the males might be chosen as mates for the various females, because those few males have whatever it is to attract the female. So again, you can get a change for whatever those traits are that are attracting the female, and get a change in gene frequency, so more and more of those particular traits that are attracting the female start showing up in the population. So let me say something about that. Something about that. So let's say if females choose a mate for particular characteristics, for particular phenotypic expression or whatever, and the phenotypic expression in this case would be behavior that say that attracts the mate, or fancy feathers or fancy plumage or whatever. So if females choose a mate for particular traits, these the genes for these traits can increase in the population. And then you could say, well, then won't that kind of make it that those traits are going to be expressed even more and more and more? Yeah, that might be the case. And if you want to see some incredible examples, presumably this is what happened. We don't know absolutely about it, but they have fossils. In fact, it's not even that long ago. The Irish elk. The Irish elk has these big antlers. Well, I mean, elk already, we see the
they're huge, right there.
These huge antlers. I almost got a taste of them once in my backside. I was quickly got behind a fence, but anyway, but if you ever look at some pictures of the Irish elk, they're no longer alive. But over time, because they have all the skeletons of these things over time, their antlers got bigger and bigger and bigger and bigger. And the last Irish elks that were around, the antlers were enormous. And presumably the larger the antlers, the more of the male, the more likely they were to attract a female. And there was what you would call selection pressure for the antlers to get bigger and bigger. And then there's got to be some point at which, there's too much metabolic effort put out to make those huge antlers, and it's got to be a
Eventually
negative
their
effect
head,
after a while because they're
yeah.
working so hard to make these big antlers. How are they going to be able to feed and eat and everything, and not spending enough time doing something like that? Whatever. I mean, that's the only thing we can guess because the Irish elk did die off, and I don't think they necessarily died off by being killed by humans. I think they died off on their own, and it may just have been because of this incredible selection pressure.
That size matters.
Yeah.
How long,
Louder, I'm sorry.
how long before they start doing the wheel there?
I know. They're
Yeah,
so heavy.
yeah.
That's
Back
right.
and it's just
Yeah,
uplift.
no, it's a possibility. So anyway, so that's kind of an interesting idea, but still, nonetheless, the main idea is that with sexual selection, it can change. Again, what are we talking about? Changing the gene frequency because the female
Over a short time.
is mating with males that have particular genes, so those genes go on ice. And that's all we're talking about in microevolution. The fifth one, the main, and this is the main cause, natural selection. We talked about that last time, but this is the main cause of microevolution. Main cause, or main agent, I should say, not cause, main agent of microevolution. So the environment, of course, is selecting for those individuals that have those traits that are most likely to allow them to most likely survive and produce and all that. So we've gone over that already, so I'll go over it more. I do want to talk about a couple of things about natural selection that we didn't talk about. Okay, so I'll come over here and erase this. Just because it has some interesting things to think about.
Finally lost a guy.
So three modes of natural selection. So I'm going to put all three down and we'll just talk about each one real quickly. I'm not expecting you just to say, oh God, mode 1, mode 2, mode 3, but just I want you to think about it. What they're called. One is called stabilizing selection. Stabilizing selection. One is called directional selection. And it's called disruptive selection. Now, I'll just give you examples. Stabilizing selection is basically saying that the extremes are not so successful, but some kind of in between is more successful. So the extremes of whatever the phenotypic expression is. Okay, we're not saying what it is. I'll give you an example, but stabilizing selection, the extremes are not the extreme. When I say the extremes, I'm talking about some phenotypic expression. The extremes are not as they don't survive as well. They're not as
favorable.
Viable. There you go. They're not as viable as the in between. I'll call them the in betweens. How about that? The in betweens, the more like the mean as opposed to the extremes. Okay, but I'll give you an example. In humans, we're talking birth weight. Birth weight. The very large babies and very small babies are not as viable, right? the in between sized babies are much more viable, much more likely to survive. But a really small baby, they have a much higher death rate. Very large babies, they usually indicate there's something wrong. So birth weight in humans sort of represents that. So I'll say, for example, the birth weight in humans. Okay, directional selection is where one extreme is more preferential. And there's an advantage in whichever the extreme could be extreme for small or big or whatever it is, but in this case, one extreme is more successful, we'll say. And that seems to be the case in the development of humans in terms of the human brain. The human brain, over a relatively short period of time, got larger and larger and larger. And so we have a large brain, relatively speaking. Certainly, compared to other animals, certainly compared to other primates or whatever, we have a large brain for our size.
And it's in our head.
When they look at the skulls of some of the proto-humans and look at the size of the skull, there was definitely seemed to be the case, we'll put it that way, that there was an advantage in having a larger brain. And so the individuals with the larger brain seem to be more successful and reproduce more, and so the brains got larger in these organisms. Now, we say this because we kind of liken it to the number of transistors on your little
computer,
CPU,
the IC,
what's
and the on-off switch and binary.
So
Yeah.
the larger the brain, the more capacity you have for dealing with the world and therefore surviving. That's not an unreasonable thing to say. A larger brain by itself doesn't necessarily mean that's what it's going to be, but certainly as humans, and that it seems to be pretty important. Okay, but anyway, disruptive selection means that both extremes might be favored, but the middle may not be, depending upon. We're just talking about these are examples. Both extremes might be favored. Over the middle, the in-between, over the in-between. Again, I'm saying it this way, it's going to be a little bit funny by the way I say it, in-betweens. And one example to give you on this, and if you thought about it, you could maybe come up with some examples too, but I know that in Africa, there's a certain finch, there are two types of finches, and one type has a really small beak and one type has a really large beak. But there aren't, for this particular kind of finch, there aren't any in-between size beaks. There's something about, one with a small beak, eats one kind of seed and it can open the seed just right and chew on it or whatever it does. The other one, the long beak can get to a certain other kind of seed and get to it and eat it. But in between, they can't eat the small things and they can't get to the big things, what the big things get to. So anyway, for whatever reason, the extremes are better.
Yeah, that's
For this particular situation.
Isn't that the kind of what they Darwin hypothesized about some kind of organism must have a really long tongue to feed on the bottom pollen at the bottom of that one type of orchid, and then they just discovered it.
That may be, yeah.
They discovered it.
And
it exists.
Yeah.
He was right.
Yeah, that's probably true. Yeah. I can't remember that in detail, but you probably.
Yeah.
Yeah. Louder. I'm sorry, louder.
Would some species of spiders be a good example of this? Like how the females are super huge, but then the males are really small.
Yeah, that's a little different because it gets to the sexual selection thing, but there where the females tend to eat the males.
Yeah, so they're easier when they're small. They don't have to fight
and
them.
And the males are eaten, but yeah, that's an example of sexual selection. Yeah.
Sexual dimorphism is important in spiders because females go gravid, which means they carry the eggs in their body.
Yeah.
Oh yeah, right. Yeah, so that makes sense. Yeah.
So, but that's more of a sexual selection thing, I think. Yeah. Let's have a couple minutes. Let's talk briefly Oh boy, briefly. Yeah, we'll talk briefly about macroevolution. Let's do a little bit of macroevolution. And we'll spend a little more time on this next time too. And macroevolution, remember, was talking about formation of new species. Just because you're changing the gene frequency doesn't necessarily mean you're changing species. We're still talking about all these whatever organisms we're talking about here. They're still the same species. But when we're talking about macroevolution, we're talking about new species developing or evolving. Now, obviously, as I said before, this is going to take a long time. This doesn't happen in a short time. First of all, let's talk about a species. Let's sort of give a definition of species. It's a little fuzzy concept. I mean, it's relatively straightforward, but it's not an exact concept. It's not something you can say absolutely this is this species is different from this other species. So the way we usually speak of a species as potentially interbreeding groups of some population. Potentially interbreeding groups of a population which are reproductively isolated from other groups. Now we're going to come back to reproductive isolation, and I don't think we'll have time today necessarily, Mike, which are reproductively isolated.
I have a question about Neanderthals and humans. They're considered two different species because they could interbreed.
So can horses and donkeys.
Yeah, so here's the deal. But here's the deal. And this gets to be part of the species concept. As I say, potentially interbreeding. Now, generally speaking, that the horse and the ass don't interbreed, but they can, and we can do it. Or we can make them breed, or artificially make them breed, and we do get offspring, but they're not viable. I mean, they're not.
They're sterile.
They're sterile. They can't. They themselves can't. So we would put them in separate categories, separate species, because their offspring are sterile. Human, we think for various reasons that probably the modern human and Neanderthal did interbreed, probably. And you can, have these genetic things done now.
They did. I had a
7%
roommate.
of Neanderthal genes. Some of us have more than that, but whatever. And actually, I want to talk about Neanderthals a little bit, or Neanderthals, because we think of them as these brutish animals and stuff, or proto-humans, but we'll talk about that. But they probably did interbreed. They probably did, and we probably do have some Neanderthal genes because they're not that different. They're pretty close. But I don't know. But no one knows it for sure, just because we don't
have a set of.
We don't have DNA from the entire Neanderthal. We have some.
But they were a different species. I'm pretty sure they are.
Well, they usually see. Here's what they do. They make what they do now. And we were going to get this with taxonomy, but we're not going to get it today. But we, meaning modern humans, are called Homo sapiens sapiens. And the Neanderthal, they say, are Homo sapiens neanderthalensis, which means they're in the same species.
Different genus.
No, same genus, same species, but maybe it's different subspecies.
Yeah.
So potentially they are interbreeding, and it could be that they interbred. It may be. See, we don't know what it was like with the modern humans, and they did interact. Did the modern humans, were they more efficient at killing off the Neanderthals, and that's why they died off about 38,000 years ago, or whatever the number is, or did there a certain amount of interbreed, or was there some reason why they didn't want to interbreed?
Well, some things are so easy, caveman can do it, but a Neanderthal couldn't.
So okay, we'll finish some of this. We'll do the macro evolution next time.
Yeah. All right.
Thank you, sir.
All right. I kind of screwed up this video. I don't know if I can actually edit those pieces. See, this is why you people need to show up to class.